This is AP Biology's opening to our chemistry unit, and yes, chemistry is a big part of biology, and my favorite place to eat is C. Hopkins Mighty Good Cafe, and that's actually a mnemonic. C. Hopkins, C for carbon. Carbon is the granddaddy of them all. And then Hopkins, hydrogen, oxygen, phosphorus, potassium, iodine, nitrogen, sulfur. Hopkins. Mighty Good, Mg, magnesium. Cafe, Ca, calcium, Fe, iron. C. Hopkins Mighty Good Cafe, a great place to eat. You get all your essential nutrients there. Those essential nutrients, those essential atoms that bond to make all the biomolecules are the foundations of biology. Every process in biology is based on chemistry. Interestingly enough, chemistry does not work sentiently. Chemistry doesn't work by decision or choice, but rather by the laws of thermodynamics. So if you're going to track something in biology, you can often look at the thermodynamics of the chemistry, and it will help you understand what's going on. So take, for example, the Krebs cycle, the cycle that basically brings the energy from a sugar molecule into a whole series of steps where energy that can be transferred with some efficiency into small molecules of ATP. If we want to understand something that's happening in a living cell, what we really want to do is figure out the chemical processes and basically follow the trail of the energy. So I'm going to do a quick chemical equation, and this is going to have a reactant side, and then it's going to have a product side. And this is a classic of biology. This is how energy is transferred from a molecule of sugar basically into the high energy bonds of adenosine triphosphate. But when you see the equation, most everything's left out, so just bear with me. We have C6H12O6, and this is sugar, right? Glucose. And then we're going to add 6 oxygen to that. And this is, oxygen gas. And then we have our chemical process arrow here, right? This is our chemical reaction. And on the product side, so this is the reactants here. We have the reactants are sugar, C6H12O6, plus oxygen, 6 oxygen, 6 O2. And then we're going to get on the product side, we're going to get 6 carbon dioxide. That's from breaking down through basically oxidation of the sugar molecule and taking the energy out of those bonds. And then we're going to get 6 water. And where the water comes from is really pretty fascinating, so I'll just break it down really fast. You've got an oxygen from right over here, and you breathe in the oxygen, and now the oxygen's made this way through the bloodstream and has found its way to a cell. When you're oxidizing that sugar molecule, at some point, the oxygen becomes the electron acceptor. 2 electrons from your busted down sugar process are going to find their way to the 2 open bonding sites in the outer region of the oxygen atom. In doing so, protons will follow. Basically, if you have a proton and an electron, what you have is a hydrogen. And so what are we doing? We're adding 2 hydrogens to the oxygen. By the oxygen being electron acceptor and the protons following the electrons, we get water. This is, of course, where we also are going to say we have energy. Bam! And some people say it's 36 ATP, some 34 ATP, some 38 ATP. It's just going to be a lot of ATP. And the process is not 100% efficient, and we're not always using exactly the same conditions. So that number's going to vary. All right, this part right here, though, this is of interest. We're going to make water, right? H2O. H-O-H. So you can have this process happen quite a bit. Over the course of respiration, you're going to make water. But every once in a while, we're going to make, water. What happens when you have an oxygen bonded to an oxygen? Well, you can still receive 2 electrons and 2 protons, but this is not water. This is hydrogen peroxide. And if you've ever read the caution label on a bottle of hydrogen peroxide, you know that that's not good stuff. On 3% hydrogen peroxide, it's still something you don't want to drink. Making hydrogen peroxide could be detrimental to the health of your cells. So we need another process to break down peroxide. Now, if you look at hydrogen peroxide, and we'll look at some paper clips dropped in peroxide, it's very unstable. Just by putting something in a paper clip, now you see the little bubbles. What we've done is we've basically created a situation where the unstable molecule now has a good reason to break up. Well, hydrogen peroxide, I like to call oops water, OO, oops. Hydrogen peroxide has to be broken down safely. And so what we want to do is break it down. When it breaks down into H2O and O. And if you have two of those, then what's going to happen is these two oxygens are going to come together and become diatomic oxygen, O2. Go through the thermodynamic processes of respiration and get energy from the process. Get carbon dioxide and water as your products. Breathe out the carbon dioxide. But then, whenever you have a problem, oops, use another enzyme, a liver enzyme, to break the peroxide down quickly into harmless water and oxygen. And the oxygen then can form oxygen gas. Let's see that happen for real. So in short, basically, the liver has enzymes. These are proteins that serve as a place for the chemical reaction to happen more efficiently. Enzymes are not part of the chemical reaction. All they do is lower the activation energy needed. It's kind of like a key in a lock. When the key, which would be like the chemical substrate that's going to react, fits the lock, it turns. When an H2O2 finds the binding site, the active site of the enzyme, it lowers the activation energy for that reaction to occur much quicker. So by having an enzyme in the liver that has a docking site, an active site for hydrogen peroxide, and lowers the activation energy for the peroxide to break, what happens is we rapidly produce oxygen gas and water from hydrogen peroxide, and that detoxifies the blood. When those bubbles are rising, one thing my students often notice if they're holding the beaker is it gets very warm on the bottom. Where's that warmth come from? It comes from the breaking of a chemical bond and the release of energy. Remember that energy is stored in chemical bonds. So if you need some proof as to what's going on here, is that really pure oxygen? I can show you and assure you that it is indeed pure oxygen, because pure oxygen is, pure oxygen is what we need for carbohydrates to go through combustion, correct? So now I'll get a match. This is what a fire burns like at 21% oxygen, and here's what it is at 100%. Ooh, and that went pretty fast. It doesn't want to go down, does it? Let's try again. 21%, 100%. We're going to be covering proteins, but just scratching the surface. We'll cover this in much more depth a little later on. Today I want to share with you the concept about form and function in proteins as it relates to the way that proteins are built of amino acids. So an amino acid basically is a carbon spine with a hydrogen, and then a carbon with a double bonded oxygen and a hydroxyl, and that's called a carboxyl group, and then an amino group, which is an NH2, and then right down here we're going to have some kind of variable group, and the variable groups are biomolecules. Maybe they're some kind of sugar base or something like that. But this is a variable group. One example of many, 20 different variable groups actually. We have 20 different amino acids regularly found in nature, and all proteins are made of just 20 different amino acids, which, when you think about it, isn't too limiting. We have only 26 letters in the English language, but we have entire dictionaries made of very short words with different combinations of just 26 letters. So if you can put 1,000 of these or 10,000 of these together, and you can use 20 different amino acids to do so, well, the number of proteins you can come up with is pretty endless. So I'm going to just make my variable groups have personality. So I'm going to draw another amino acid here, and I'm going to show you how we bond these. So here's my next amino group, and my carbon spine, and my next carboxyl group. And let's give this one a sad personality. And this one is going to lend a hand to this one. And we'll get to that in a little while. The next thing that's going to happen is something called dehydration synthesis. And another way of thinking about it is, because water is going to appear, we call this condensation reaction. And when it occurs between two amino acids, we call it a peptide bond. But wait, what is this thing? What's going to happen? My carboxyl group is going to get rid of an OH, and my amino group is going to get rid of an H. That's where the water comes from. Water appears. Dehydration synthesis. Well, we haven't synthesized anything yet. I have an empty bonding space here and an empty bonding space here. We're going to bond covalently by sharing electrons and sort of fulfilling the needs that the carbon and nitrogen now have. When that happens, they will bond together. The geometry is going to change a little bit. Our double bonded oxygen is going to perk up a little bit, and our hydrogen is going to perk up a little bit. And now we've just bonded two amino acids. And when these two variable groups attract, they're going to cause a kink right here. So there's actually a level of organization in proteins. Your primary structure is the order of different amino acids that you have. Your secondary structure is going to happen with the gentle kink that happens when you have this double bonded oxygen and this hydrogen up here, and the interactions between electrons. There's a molecular geometry that's going to happen. It's going to give a little kink. That's called secondary structure. But the third level of structure, tertiary structure, occurs right here, where our variable groups are going to interact. There's acids and bases. There's positive and negatively charged, polar and nonpolar. There's a lot of types of interactions. If you have two nonpolar molecules, right? It could be very much a straight line, but we're going to get kinks and twists. If you start putting a bunch of amino acids together, you might start getting kinks and twists around that start to make a weird shape, and maybe you even end up with something like a globular protein that has a whole bunch of twisted and shaped layers. And that's where it gets interesting. Somewhere, how about right here, like a little cup, you might have something that perfectly fits with some kind of molecular substrate out there in nature. Hydrogen peroxide might fit perfectly right there. So this is like the lock, and hydrogen peroxide is like the key. And this lock and key hypothesis shows how the chemical substrate could fit right in here perfectly, and that's going to lower the activation energy and allow this enzyme to catalyze a chemical reaction. This globular molecule, it's an enzyme called Catalase that in animals, for example, deals with breaking down hydrogen peroxide, can catalyze, I think it's 18 million events per second, but I'd have to look that up. I think that's about right. So from the bond between a carboxyl group and an amino group, the kicking out of a water, we can keep doing this over and over and over, because over here I could bond another carboxyl group, and over here I could bond another amino group. Build these long chains. These chains then go through their tertiary structure interactions with the variable groups, and they make a shape, and that shape has a function. Now, if I was to subject this to acid or a strong base, it would interact with some of the variable groups. For example, if this right here has an OH group on it, and I was to take a strong acid and subject that acid to this, I could neutralize that. And if I neutralize this, I'm going to change the personality. When I change the personality, I change its interaction with its neighbor. When I change the interaction with its neighbor, I change the shape. And that changing of shape is called denaturation. So I would denature this protein. I would remove its nature. I would remove its way of being in its environment. So denaturing a protein can be done chemically. It can also be done with heat. What we're going to do next is look at how denaturing a protein would affect a liver enzyme, catalase, in its ability to catalyze the breakdown of hydrogen peroxide. All right, so I'm going to share with you a little bit about the denaturation of the liver enzyme catalase. And so we're going to demonstrate with 100 milliliters of hydrogen peroxide in one beaker. All right, so I have 100 milliliters of hydrogen peroxide in both beakers. What I'm going to do now is I'm going to take some deer liver, and I'm going to try to make the pieces pretty similar. They're both going to be about the same size, and they're both going to have some of the same attributes to them. I'm just going to use my fingers. So both pieces are going to be from the same cut, and you can see that there's this area that hasn't been oxidized at all, really nice and fresh, and I'm going to cut it right in half. And, whoopsie, one half I'm just going to drop right in the peroxide. And of course we know the expected result. The peroxidase is now rapidly ripping apart those peroxides. Well, not really, it's lowering the activation energy and allowing the peroxides to separate out into water and oxygen. This other piece right here, I'm going to put in here. And just hold on for a second. A lot of oxygen being produced. So from the 100 milliliters of peroxide, we know that there is a known amount of reactant. Eventually we're going to have water and all the oxygen that it took to use up the 100 milliliters of substrate. The enzyme is not involved in the reaction as a reactant, right? It's just there participating by lowering the activation energy. So the enzyme is not consumed. It's not used. Wow, look at that bubble right there. I really wish I had a match. That would have been fun to flame that bubble. But this is pure oxygen evolving right here. Now on the other side, I'm going to set this aside. Right here, this piece of liver, I'm going to add sulfuric acid. And so think about what you've learned now about the structure of proteins and the fact that the variable group interactions are key to the shape of the protein and how then that shape confers the function of that protein. When I add sulfuric acid to the liver, the sulfuric acid is going to be donating H pluses. A lot of the base groups are going to be neutralized. They're going to no longer have the same relationship with neighboring amino acids. So that's going to change the tertiary structure. When the tertiary structure changes, the function changes. Sulfuric acid is going in. You can see it's starting to look very clay-like. I'm going to place this liver in the peroxide now, and understand we've changed the variable groups with the sulfuric acid, right? So enzyme structure should be different. And if that's true, we may have lost our active site for the peroxide. If that's true, then when I drop it in the peroxide, nothing happens. I had a brilliant student today say, what happens if I expose new surface area? What happens if I cut the liver open to reveal an undamaged portion? Now if you look in there, it's looking pretty good in there. So let's try this. You can see that there's some enzyme, not very much enzyme left in there, but there's some enzyme left in there. that can help catalyze that reaction. Well, there you have it. The denaturation, or the changing of the structure and thus function of a protein, that can be done by acid, by strong base, by heat. The denaturation of protein changes its structure, thereby changing its function and the way it relates to its environment. By subjecting the liver to sulfuric acid, you saw the color change, so that was an indication that something chemically had changed. Its absorbance of light changed. Structurally it's different, chemically it's different. The active site might be compromised. We put it in the peroxide and sure enough, no more active site. We cut the liver open to expose some of the fresh liver, and we found there was very few enzymes still left there that hadn't been reached by the sulfuric acid. So that is protein denaturation. Hey, real quick note about safety. When I'm using a strong acid, in this case it's 2 molar sulfuric acid solution, I'm going to wear my goggles. And the reason is pretty clear, pun intended. My eyes are very clear, my corneas are transparent, and that's achieved through having many, many very fine particular proteins in parallel arrangement. If I was to denature these proteins the way I've denatured the liver protein, I would also change the shape and the function of the proteins in the fronts of my eyes. I would lose that parallel arrangement and my eyes would become opaque. So here is a white-tailed deer liver, and it's a very healthy liver. It's got good color, and I'm going to cut off a section here, and we're going to use the deer liver enzymes that will run out of that section. I'm going to label some containers for storage. Here's some deer liver. Maybe I'll have some turkey liver or goose liver as well. We can use different species and compare them. And what we're going to do is break down hydrogen peroxide. And here is my peroxide. You'll notice I'm putting 10 milliliters in each. And I've refined the measurement to have exactly 10 milliliters in each because I want to have the same amount of reactant. I want these to come to the same end point. If I have the same amount of reactant that will come to the same end point, then I can sort of blame the variance on the liver enzyme. And so what I want to do now is add, in a controlled fashion, some liver enzyme to a plate here where I can pick it up with some filter papers. And these are 1 centimeter squared filter papers. And I'll kind of dab them to get any excess drip off because I want to have the same amount of liver juice per filter paper. In this case, I'm going to put 1 filter paper kind of up on the ceiling of the reaction chamber, so it's not yet interacting with the peroxide. It's up on the ceiling there. The peroxide's down on the bottom. And I can react that when I want to simply by rolling the situation over. Now I want to put 2 filter discs in the second one, speeding up the video here just so you can get a sense of what's going on without having to sit through it all. There's 2 filter papers, and now I'm going to put 4 in the third reaction chamber. In biology, we often double each time because that order of magnitude allows us to understand and see the differences. So now I go from 1 to 2, 2 to 4, and this is my 4 filter disc reaction chamber. So when I roll these over, the peroxide is going to really quickly break down and evolve oxygen as peroxide becomes water and oxygen. And that oxygen is going to go bubbling out of the jet. And I can capture that bubbling oxygen through displacement in a graduated cylinder filled with water. So here come the bubbles. And so for 1 disc, I'm now displacing. And if I have a stopwatch or a timed video, I can actually measure the amount of time it takes to displace a certain volume of oxygen. If I take the slope, y2 minus y1 over x2 minus x1 for any one of these, where x1 and y1 are both 0, so basically y2 divided by x2, that's milliliters per second, I can come up with the rate of oxygen evolution. And then I can compare, look how fast 2 goes compared to 1. I can compare the rates mathematically. So I do this until it comes to an endpoint, but it's practical to just measure somewhere along the way when you still have a good reaction happening. You're going to see your straight line, you're increasing at an increasing rate, go to increasing at a decreasing rate, and then it's going to flatline. So in that first, there's the little bubbles. So you want to get your data in the big bubble time. Once you start getting those little fizzy bubbles, you're kind of moving into the next phase of things where there's less substrate. All right, so now let's think, what are some different types of experiments we can do using this basic idea of evolved oxygen coming from the breakdown of hydrogen peroxide? So now that you know how to measure the rate of oxygen evolving from the breakdown of hydrogen peroxide using water displacement, we can now start to design experiments that will help us understand more about the liver enzymes themselves. For example, you could try to denature the enzymes using sulfuric acid at different concentrations and see what sort of levels of damage occur with even very small concentrations. Or maybe you could look at some kind of assay with 1, 2, 4, 8 filter paper concentrations of deer and then compare that to the rates of other species to see if you can sort of tease out what concentrations of liver enzyme are found in maybe the liver of a goose or a turkey or a chicken or a duck or a squirrel. Another option is that you could actually vary the amount of peroxide. Now remember that 30% hydrogen peroxide is dangerous and you should be fully equipped with lab protective gear. If it's dangerous to you, what do you suppose that does to liver? So there's a lot of experimental variation you can do, and you can also team up with other scientists and figure out what your results mean and what their results mean and how those results might speak to each other. A couple of parting thoughts. Vocabulary. Anything that ends in ASE, I think of that as the best card in your biological hand. That's your ace. ACEs are enzymes. So for example, Catalase is the enzyme in your liver that breaks down hydrogen peroxide. Amylase is the enzyme that breaks down amylose or starch. It's in your saliva. It helps you pre-digest your carbohydrates. Helicase is the enzyme that untwists the double helix of DNA. So it's a helicase because it works on the helix. Some more vocabulary. Remember, every enzyme has an active site. The active site is the part of the enzyme that fits with the chemistry. The fit with the chemistry, that is where the active site meets the substrate. The substrate is the chemistry that is going to happen. So there's a couple of different types of substrate interactions that can occur at an active site. One is where two things come together and with lower energy are able to be combined, and the other is where one thing enters the active site and is more readily broken apart. So enzymes are both builders and breakers, and so that occurs at the active site. The enzyme lowers the activation energy, the amount of energy required for the chemical reaction to occur, but it is not involved in the chemical reaction itself. Enzymes are not consumed by their actions. If we're doing a liver lab and we start to see destruction of the liver, chances are it's because of the things that we're immersing the liver into, and remember this is not a living, healing, and repairing liver, but the things that we're immersing the liver in might actually be caustic or destructive. So, for example, 30% hydrogen peroxide or sulfuric acid. The liver's not involved in the chemical reaction, but it sure is taking a beating. In the normal life processes, the liver is not involved in the chemical reaction in terms of being consumed. It simply provides the active site in which the activation energy is lowered and the reaction can begin. So some ways to think about it. I hope that's useful. And this has been your liver enzyme lab tutorial. Hopefully I'll continue this thought a little bit later in another video and maybe improve on the one we have. Take care.